thank you everyone for the opportunity to speak, Jesse and all the organizers and folks on the Columbia. And obviously things here in La Jolla are a little different than how they have been in New York City. And first and foremost, I want to thank everyone, not only on this call, but also participating and most importantly, responding to this pandemic in your city, as well as obviously every other part of the world that has been affected. I wanted to share a little bit of work and a few slides around the work that we've done in drug repurposing. This has been work that we started essentially now back in late January when we were first approached by folks in Hong Kong University to make drug screening libraries available to them, as they were first isolating virus from patients who had traveled to Wuhan and had come back to Hong Kong and were displaying symptoms of the infection. And so from that work to where we are now, which is about 5 months, I'm happy to report that we've done almost 24 different screens, both cellular screens and target-based drug screens, and have generated a large amount of information that's all publicly accessible for folks to be able to see kind of our work in drug repurposing. That is really a global effort, but one that started by kind of building a really what we believe to be a best-in-class drug repurposing library for not only this infection, but we've used it now for several infections and initiated clinical trials based on that. So I'm going to share a few slides around our work. I hope everyone can see that okay. So, you know, kind of where were we at? Obviously, in before this pandemic, we had actually worked with the Gates Foundation to put together a compound collection of about 12,000 different molecules, really based on the premise to essentially, as much as possible, democratize the process of drug discovery. What do I mean by that as a chemist? You know, I've been doing drug discovery chemistry for about 18 years. And, you know, one of the real challenges between working in the private sector, and I was at Novartis for about 9 years, and then the last 8 years at Scripps at a nonprofit, is this concept that, you know, you don't need to know a chemist or a pharmacist to be able to do drug discovery for small molecules. The hope was that clinicians, biologists, pharmacologists around the world and in many different areas of science can approach and address a compound library without having to ask their neighbor to provide drugs to them, but actually be able to access a library that is open source and free to access for a variety of different disease indications, particularly ones that are impactful in low and middle income countries. I realize COVID-19 is one that goes well beyond this range of low and middle income countries, but one of the key aspects of what we wanted to do was to make the drug collection available to folks. We've actually sent screening equipment, we've sent postdocs. We actually had a postdoc go to Mae Sot in Thailand for 3 months to screen the ReFRAME collection on dormant stages of malaria, vivax parasite, for example, where, you know, being able to move the assay would be impossible. And so we actually sent resources and people to be able to conduct that screen. And in many ways, when we sent plate readers in this compound collection to Hong Kong at the end of January to address this particular infection. So we've taken a very broad approach to this. 12,000 compound drug screening library nowadays is considered to be a small collection and something that many different groups, various certainly for-profit groups that screen millions of compounds, but also academic and nonprofit groups, including folks at Columbia as well as here at Scripps and around the world, can access, you know, a set of 40 or so 384 well plates and screen compounds relatively quickly in a variety of different assays. So we've actually went through the literature and identified nearly 14,000 compounds that have some level of annotation of being dosed in humans with some level of safety data. We've gone into the lab and synthesized 6,000 compounds. That was a significant chemistry effort. At one point, we were actually using, through contract research organizations, 500 plus chemists to synthesize these molecules. at 20 milligram scale and make them available across the screens that we're doing. The good news is now in the days of kind of acoustic dispense and miniaturization of assays, 20 milligrams can last us about 10,000 screens. And this is some work we published in 2018 when we first published this collection. The really interesting part is, in fact, half of this collection are molecules you can purchase from vendors around the world who sell preclinical drug screening collections. So we actually had to make these compounds. And as I mentioned, we send the collection, we send plate readers, we send people as needed to be able to do things. So what have we been able to do? So in the last kind of 4 to 5 months, we've been able to run about 20 different screens that have been completed, several that have been run at Scripps, but many others that have been run throughout the world. We've made that data publicly available. We've actually identified a set of about, I'd now say about 60 or so bona fide compounds that do show antiviral activity, both in viro cells, as well as in human cells, as well as now in kind of primary systems, primary cell systems, to be able to demonstrate that the activity is kind of, conserved across multiple different groups. I think in the early days, obviously, there was a lot of people working in parallel. A lot of clinical trials were being initiated, both in the Far East as well as throughout the world. And so we've tried to make some sense of kind of which compounds kind of have that kind of bona fide set of properties that make it particularly interesting as a drug repurposing opportunity. That includes obviously some approved molecules, but we also see a large number of molecules that made it into the clinic that were compounds that are not commercially available, but were stopped for some reason or another, either commercial reasons, most often commercial reasons. And then again, obviously, I don't have to speak to this audience about the value of drug combinations. Clearly, we need to be thinking about not just nucleoside and polymerase activity, but protease activity, a combination of that with host factors to be able to address not only the virological component, but also the inflammatory component of this disease. So, drug discovery is hard. I just really have this as a background slide just to show the attrition of projects. And so being able to get to a molecule and optimize it to a drug candidate takes a lot of work. And we believe that the ReFRAME collection tries to recapitulate many, many decades of drug development work into generating molecules that one can repurpose safely. As I said here, we see here, this is analysis done by AstraZeneca several years ago, but this is often a cause not only of efficacy being a reason for failure of drugs, but even for later stage molecules, commercial reasons and strategic reasons why molecules fail. And so being able to understand if those compounds can be repurposed is critical. And I think that's really quite challenging to be able to get all of these together, even without a pandemic on our hands. At Scripps, we were essentially closed the labs for 2 months here in La Jolla, where we were only doing work in the BSL-3 around COVID infection and related work. So, being able to do that is hard enough, let alone kind of the situation that the world is kind of in now. And really the ReFRAME collection starts with a variety of different, starting points. There's this idea of large screening libraries to, known ligands that have been published in the literature to natural products to kind of repurposing. So while it's a long continuum of the kind of drug sort of products that might be available, antivirals particularly have been challenged, right? Nucleosides, protease inhibitors, et cetera. We don't have the broad level of kind of druggable chemical space in the antiviral world the way that we do even for bacteria and parasites. And so finding the right starting points and then most importantly finding the right repurposed drugs is a real challenge. So we think that this collection kind of addresses an attempt at a first approximation to be able to get to a workable set of molecules that can be shared with the community to actually consider how we can move those forward. So I'll talk a little bit about the ReFRAME repurposing opportunity program. So this is really, the goal of it is direct repurposing. And so we've actually repurposed 2 drugs, one for cryptosporidium and the use of clofazimine, which is used for leprosy infection. So moving an antibacterial into an antiparasitic drug indication, as well as oranafin for treatment of XDR and MDR TB. So those 2 trials are ongoing. One, the TB trials in South Africa, the crypto trial is in Malawi. And again, was work done by screening the ReFRAME collection against, new pathogens or pathogens that these compounds may not have been tested against, and then repurpose them. We also see an important aspect of not just repurposing and rescuing compounds that could be used for another infection, but also this idea of accelerated medchem. So I think we've done this now in the context of antifibrotics and a few other areas as well. We're having a hit, and we published this a couple years ago on the use of voriconazole, for example, as an antifibrotic. One could actually take a molecule that has a significant amount of efficacy and, most importantly, safety information around a molecule and be able to derivatize it such that the molecule can go from a acute indication use like itraconazole is for fungal infections and be able to turn that into a safer molecule to be used in chronic conditions, for example, for IPF or NASH. And so in that case, the medchem from a chemist perspective, the range of parameters you're trying to fix of a molecule to be able to make it more amenable for use in a chronic indication or in a different disease setting is certainly advanced with a molecule where we know a lot about the pharmacology and safety of it. So again, ReFRAME is about 12,000 different molecules. It contains some preclinical molecules, particularly ones that have been through efficacy studies in rodents and non-rodents. But most importantly, the majority of the molecules are compounds that have been characterized, small molecules, less than 700 Daltons that have been put into humans. And again, we did this work with about $20 million worth of support from the Gates Foundation to put this collection together. What this allows us to do, I won't belabor this point, is to move quickly into potential phase 2 studies, as we've seen with regulatory authorities, not just with FDA, but around the world. The ability to initiate phase 2 proof of concept studies with multiple arm proof of concepts in the COVID infection-19 situation has obviously been accelerated, and certainly, one can take it leads and further optimize them for be able to be more specific to this particular infection, which essentially was work that was, being initiated back in January by folks at Gilead around remdesivir. So, how can ReFRAME have an impact? We obviously think drug combinations are critical. As I mentioned, we started this work in late January. We shared the collection within days of a request from Hong Kong. So our MTA is exceptionally straightforward. All that it asks is that people publish as soon as hits are found and they're unblinded, that they are publicly made accessible within 30 days. So it is a very straightforward approach, and we think that is something that could be really valuable. We've also developed assays ourselves, which I'll talk about in the next few slides, around actually repurposing drugs using a first high-throughput HeLa ACE2 cell system, where we've actually identified a large number of molecules that we think are complementary to some of the work that have been done in Vero cells, including some of the work that we supported in Hong Kong as well. And then also, one of the things that we've done is, be able to think about kind of, disease prevention, right? So in a prophylactic sort of setting, what are the right sort of ways to be able to get to, at-risk populations, not only in the US, but, also part of our mission statement is in low and middle income countries about being able to get safe oral medications that can be given as prophylaxis, early in treatment, mildly symptomatic patients, and be able to protect their communities as well. So we've done some work, particularly in HIV and malaria. I don't get a chance to talk about that today, about kind of long-term chemoprophylaxis and long-acting chemoprophylaxis. But that's certainly one way we're thinking about this, is that there's a prophylactic component of this that's critical to be able to really be able to reduce infection rates. So, what we've been able to do, again, as a working model, is once an MTA is executed, we provide acoustic dispense pre-spotted assay-ready plates of these 12,000 compounds to folks around the world. Intellectual property is actually the investigator's ownership. Again, all that we care about is being able to take this annotated drug screening data and put it into a public portal, which we call reframedb.org, which I'll get to later. And again, eventually there are terms around being able to provide drug product, to throughout the world to be able to kind of really maximize the findings from screening of this library. And again, we've also provided smaller parts of this collection. We've screened this collection against worms and things that only work in 24-well plates. So we're happy to provide smaller parts of this collection. In fact, we have around 170 or so molecules we call kind of a focused antiviral set. But our preference is for folks, if they're possible, to screen all 12,000, because we found really interesting things beyond what the literature has kind of described over the last 6 months around what potentially could be useful small molecule therapeutics for COVID-19. The composition of this library, again, in the process of kind of making this an open access platform with this compound collection is actually, not surprisingly, a lot of experimental molecules, things that are not approved throughout the world. One thing I will mention, we spent a lot of time is trying to figure out what drug databases to actually use to be able to put this collection together. And so what we have found, in fact, is that based on editorial standards, we actually needed a minimum of 4 different commercial drug databases to be able to put them together, because we saw a lot of non-overlapping, different ways of editorializing whether or not a molecule has actually been safely put into a human trial. So taking one specific drug source, drug data source, and saying that this is the right answer actually doesn't get as much coverage in terms of the chemical matter that you want to be able to put into a collection. So we've done that, and not surprisingly, many of these drugs have been developed for everything other than anti-infectives, given how the industry, especially the commercial industry, has moved away from anti-infective drug discovery. This collection has been screened in the first 3 years. Obviously, now we've essentially doubled the screening data around ReFRAME just in the last 4 months with COVID-19. Prior to COVID-19, we did do a fair number of screens, both internally and externally, around a variety of different work. We published our first COVID screen in the middle of April, which was the screen that I mentioned was initiated in Hong Kong in February. And then we've also been able to screen a variety of other different pathogens. And again, really the key aspect of all of this is being able to reach to the neglected tropical disease, I'll just say the neglected disease field, and make this compound collection available to investigators throughout the world. We have a few examples of things where we've actually been able to directly repurpose. I mentioned uranafin and clofazimine that have already entered trials. We have several other things that look very interesting that are compounds that have been either used in animal health, and so our drug collection includes many animal health drugs. We know for COVID-19 infection, particularly feline coronaviruses is an important area to make sure that drug activity in those sort of related viruses are kind of recapitulated. So animal health, human health drugs in here, and as well as potential earlier stage molecules, of which we've published most of this work just in the last 12 to 18 months around some of these other opportunities. So for the Gates Foundation, we've served as kind of a key drug discovery hub for drug repurposing, not only providing the collection to folks, but also develop internally assays within the Scripps Calibr community. And just as a background, Calibr serves as a operating division of Scripps Research. We were an independent nonprofit merged into Scripps at the end of 2018 under the leadership of Peter Schultz. And in that arena, the 150 or so people that are at Calibr serve as the drug discovery arm of Scripps Research, but we also obviously work with collaborators around the world, particularly other academic and nonprofit groups. And so this is a constellation of the folks that we've provided the collection to. As I mentioned, we started our work in Hong Kong back in late January, early February, and not surprising to this audience, I won't belabor the point that there are multiple different approaches to be able to deal with SARS coronavirus 2. I just mentioning the viral replication cycle, not even thinking about the other half of this, which is the anti-inflammatory components. So as I mentioned, we've distributed the collection to about 25 different groups. So about 20 of the screens have actually been done. The data has been uploaded. We've worked very closely with Tom Rogers at Scripps in the immunology department and developed a 3D4-well assay back in March and April when we executed the screen around a HeLa cell line that was actually permissive to infection expressing human ACE2 receptor. In fact, we found the majority of very interesting drug repurposing opportunities from that screen. We've now down selected on those hits through Calu-3s, organoids, primary lung cultures, ALI cultures, et cetera, to kind of start getting a fingerprint on which compounds look the most interesting from a broad range of activity against a variety of different assays. I think it's important to point out that while many in the field, including folks in the for-profit space that we've worked with, who've said, we don't have yet the right assays to be the definitive assay to figure out what to do because we're developing assays as we're screening drugs. While I take that point, I think it's important to point out that compounds that work in multiple different assays, and remdesivir as an example, does work in many different assays for antiviral activity on COVID infection, that there is some correlation. We can start building greater confidence in particular drugs as candidates for phase 2 studies by looking at the data together rather than just waiting for the ultimate assay that answers the question. In pharma often, including my time at Novartis, validation of antiviral assays can take months, sometimes even years to be able to get to the point where it's a definitive assay. We simply don't have that luxury here. So we need to be able to take fingerprints of compounds against multiple different target and cell-based assays to really understand what we can potentially do. We've done some work on passive antibodies and vaccine work, both with the Burton Lab and others at Scripps Scripps. I won't get a chance to talk about that today. I'm really going to focus on kind of our small molecule work. So getting to the collection, people can make requests for this collection, very straightforward. We looked at it, we internally look at all of this. In fact, we've actually allowed, unlike in other circumstances, duplication of assays that are highly similar, because we do believe that as people are developing assays and optimizing them, that the drug information we'll get from screens will be similar in some cases between assays that are highly related, but they may be different. So we appreciate the fact that there will be some duplication in the assays of many labs that are going to be running the ReFRAME collection. As I mentioned again, provide the collection within weeks of someone making a request. And then again, all we ask for is that the data is shared publicly. So we have an open access portal. This is one such example of it. This is actually the Vero E6 screen that was run in Hong Kong in collaboration with folks here in San Diego at the Sanford Burnham. And in this particular screen, you can see that we not only list the disease, the indication, how we actually ran the screen. This is a manuscript that's now been accepted in Nature. And then most important, again, in a world of kind of bioarchives and preprints, this data is actually uploaded in parallel to any preprint services. So, people can not only see the preprint, but they can also jump in and look at all of the specific drug screening information. And then you can see, for example, some of the hits here. This information can be downloaded, including access to chemical structures. So you can actually see at a drug level view, this was actually a protease cathepsin inhibitor we found in phase was entered in phase 1 that was found to be active. You can see it's active in this particular assay, in the Vero E6 immunofluorescence readout. You can actually also even see where we acquired the compound from. So if people are interested in following up, they can figure out where we got the compound from. And then this, most importantly, not only the chemical structure, but the drug information around where this compound was actually identified, what drug database was used, where it's being used, where it had been used, the mechanism of action, and what the current phase of development is. Obviously this is being updated on a quarterly basis. These drug data vendors have information updated daily, but we at least think this is a place where you can look at multiple different assays and look for fingerprints of particular drugs of interest to do. I talked about that on the portal. So one of the assays we ran after that Vero E6 assay that I just showed on the previous slide was actually develop a high-throughput CoV-2 assay in HeLAs expressing human ACE2. We actually ran the screen back in early April, and we've actually now run potentiation screens as well that show that particular drugs, for example, a subtherapeutic concentration of remdesivir, for example, can synergize with particular compounds in the collection. We were able to generate a screen with a relatively high Z prime of about 0.77. We've now been able to run drug combination screens, potentiation screens, et cetera, to be able to really be able to ask in a high fidelity assay system using human cells, kind of what are particular hits that have come out from this sort of a screen. And obviously the field is now advanced with additional kind of screens, but we did the screen with the whole collection and in multiple different assay formats, which I think adds some power to the ability to be able to start asking very defined questions around, what shifts potency by twofold versus assays that only can, kind of ascertain potency differences at kind of the 5 to 10-fold level. So one of the things we've done a lot of is obviously, and talked about a lot in the context of the drugs that have already been kind of appended, repurposed, is kind of understanding PK/PD relationships and understanding safe therapeutic drug levels. So we have actually been able to, not in the portal yet, but we've actually been able to get drug exposure information for about over 4,000 molecules in the ReFRAME collection. And we share that information with various investigators who've come up with hits, who want to ask a very simple question of, well, how much can I cover? So I use remdesivir as an example. This was their published data a few years ago in the context of Ebola infection, and kind of how much time over EC50 can one actually cover? What's the volume? What's the amount of active metabolites in this particular case? The green line being the triphosphate levels at the top of the graph, triphosphate levels in primaries in CD4s, and be able to understand kind of, is there an opportunity to actually be able to get this drug at safe enough levels in plasma, or most importantly in tissues relevant for being able to actually deal with this infection. So oral approved molecules, not too surprisingly, hydroxychloroquine, remdesivir were hits. We found a variety of other hits as well, including things that have been identified from various other screens as well, particularly small scale screens that have been done by groups now around the world, but initially was actually identified by small scale screens that were done in China and in the Far East. And so this is a list of kind of approved molecules. And you can also see here, we've actually listed, through our portal, all the other information, including the status, kind of what the activities are, what the safe drug levels are, et cetera, what the toxicity, and then most importantly, what's the half-life of the drugs and what are the biodistributions, so we can actually understand kind of what that pharmacokinetics relative to potency look like. We've also been able to do synergy experiments that I mentioned before, so interactions with remdesivir where molecules are either additive or not. We were actually particularly interested in protease inhibitors. We think that not only some of the kind of leading work from Pfizer and others in the protease space is going to be relevant here, but also around looking at host factors involved in activation of not just direct acting antivirals on M-Pro, but also, and not just TMPRSS2 to activate ACE2, but also in other potential host-directed proteases, either involved in endosomal maturation of the virus or from the kind of TMPRSS2 exocytic replication cycle. So we found a few very interesting protease inhibitors, several of which are ones that have been in humans for other indications, particularly some, for example, cathepsin K, which again, many of those molecules were certainly safe and efficacious. They were taken off from further development due to commercial reasons around bisphosphonates and other cathepsin compounds that were ahead of the clinic. And then we also found a variety of other sort of interesting molecules. I would not have imagined RNA splicing modulators used for the treatment of SMA would, for example, would be hits that would come out of the screen. But clearly they are active, and they're active within a range that we know that the safe drug levels could potentially be reached for these sort of molecules. So again, beyond the HeLa system, now looking at other systems, Calu-3s, primary lung organoids, ALI cultures, et cetera, is kind of where we're pushing this data to come up with a more definitive set of molecules by the end of the summer and early into the fall of compounds that can be repurposed, and including obviously some of the known sort of chemotypes that have come up from various other screens as well. And I'll belabor the point by just saying the bench is relatively deep here, but I think a lot of these molecules will fall out as we kind of profile them through other assays. But I think running a high fidelity screen initially on 12,000 molecules has given us multiple starting points, some of which have also been recapitulated by other folks, but also new molecules that have not been identified to move forward in a variety of other assays. So I mentioned the combination work. We've done this screen not only with potentiation, looking at checkerboard assays, but also looking at potentiation of subtherapeutic remdesivir as well. Found some interesting hits from that as well that we're following up on. We again think that this is important from not only a perspective of looking at single agents, but also helping prioritize potential combinations, which could be, potentially useful for looking at multiple different antivirals that could be working together. And again, this is a way of kind of, looking at, ways of combining drugs. For example, we combined a pilimod with a PIK5 inhibitor, which had been reported to be, involved in endosomal trafficking of the virus in the context of other infections, including filovirus, rhinovirus, et cetera. We found that you can actually, be able to back off both the plasma and the concentrations of both remdesivir and a pilimod to be able to get to essentially additive activity, which allows you to use lower concentrations of both drugs. We know there are limitations with remdesivir, both for even in a world where a sub-Q or an inhalation formulation is available down the road, but just simply, being able to get enough of that drug at two parts of the world where you simply, cannot be able to get that high of drug levels. Could we potentiate that by addition of a second agent? And again, much of this is now being recapitulated in animal models. It's been seen both with hamsters that are infective, ferrets, cats, non-human primates, of ways to potentially do some of these experiments in vivo. I'll leave as the final point here. I didn't think that we would be, talking about the next pandemic already, but in fact, sort of our work with the Gates Foundation last year started with the idea of being able to actually put together kind of a drug repurposing center around, a pandemic preparedness. So obviously in the middle of a pandemic, it's important to address this infection, but it's also critical to build the infrastructure, both within the nonprofit and the for-profit sector, to be able to expand on capabilities, both in drug screening, as well as in drug optimization and drug characterization, to be able to lead to our next potential starting points for the next infection. We had been very much focused on Ebola infection back in the 2014 West Africa outbreak, as well as in pandemic influenza over the last 18 months or so, but really thinking about this very broadly, working with both pharma collections, which has been so part of our work with the Gates Foundation and the CEO Roundtable that's been organized at the co-chair level within the Gates Foundation, but also with ReFRAME and other publicly accessible sources to be able to develop a network of partners to not only provide assays, screen, as well as the drug development aspects, that where we have more than just the remdesivir is our next option for the next infection. And with that, I'll leave many, many people to thank, obviously funders involved on the right-hand side. Most importantly, many, many people, I think in the front lines of dealing with this public health crisis. It's also important to point out the people, many of which are shown on the slide, Anil, Melina, Nathan, Tom, etc. Several of them, for example, Tom is a physician as well, also actually in doing rounds, but also being in the lab during the middle of this pandemic, coming in and being able to make their time and their effort available at the very front lines of developing assays and doing drug screens, as you know, the global public health community has been able to try to help all the patients, is also helping move the science forward. And I think as you know, conditions go up and down in terms of the infection, you know, I think it's important to point out that scientists' contribution to this as a global scientific community, in my mind, is unprecedented, with the amount of work and collaboration between various groups that keep the science moving forward, even though the virus may be slowing down in some parts of the world versus others. Thanks.
Arnab, this was a wonderful talk. Let's see if there are any questions. I have one of my own, so maybe I can get started. So we've done quite a lot of work in actually generating RNA sequencing signature of drug perturbations in a variety of different cells. We use that to actually predict some of the drugs that would target key processes that are induced by virus-host interaction in human cells. And the surprising thing was that we found that these processes are very, very different from cell type to cell type. And in particular, if you look at CALO3 or CACO2, or if you look at organoids, et cetera, and we've collected data from a variety of different labs, you can have even in complete inversion, like for instance, something that would be an initial, a block of replication in S phase may become completely inverted in a cancer cell, et cetera. So, and conversely, there seems to be tremendous heterogeneity in the response that you see from drugs in different cells. So I was wondering whether the kind of model that you've listed, which include, HeLa cells and CALO3, you think are really relevant, and whether we need to move towards more sort of, I would say, more conservative, maybe organotypic culture,
Sure.
Primary cell screens.
Yeah, no, I completely agree. So I think where we have kind of landed is that, those assays at some point will be available to screen many, many drugs. And I'm very confident that we will be there in months' time and not years' time. So I think that as a community, we're going to be able to get there. And I certainly do think, let me take the RNA piece of it first. So what we have done, and the Broad has actually done this, is they've run the L1000 landmark gene set against all of ReFRAME. In fact, they're in the process of making that data publicly available. So generally speaking, people will be able to see the perturbation, at least in a couple cell lines, of what the drugs actually do to gene signatures. So that's one piece of it. The second piece of it, in terms of particularly for this infection, I think is exactly that we will and are testing a variety of these smaller drug sets in systems where there is follow-on data. So for example, using nelfenavir as a kind of an example, we have seen kind of a signature of nelfenavir activity being preserved across multiple different systems, including primary cell systems. So how do you prioritize that molecule versus the other 50 or so molecules that are HIV protease inhibitors that are in the collection, to think about what to do next? That's where we're hoping, the answer lies, is that there will be some molecules that survive this absolute, likelihood that compounds will work very, very differently and work oppositely in some cases. And we see that just going from HeLas to CALU3s, which is the only thing you're changing. Well, it's an important thing you're changing, but you're essentially removing the endosomal pathway by overexpressing the receptor and allowing TMPRSS2 and the exocytic pathway to lead in for viral entry. We've seen differences. So I think that at the end, we're hopeful that we'll be able to run ReFRAME, not just against 20 assays, but maybe against 100 different assays, all relevant to COVID. And some of them may be the assays where people will look at and say, this works in an organoid culture. Why are we messing around with HeLa cells?
Yeah.
We're just not there right now as a community to be able to say that.
I want to also give a bit of a word of caution on the links data. So the links data, there are two issues that we spent a lot of time thinking of and addressing in our assays. The first one is that the concentration at which these assays are done are sort of predefined and not based on actual drug response curves.
Absolutely.
And so
Absolutely.
every RNA-seq profile we generate is done actually at the highest sublethal concentration that you can achieve, because otherwise you start observing processes that have nothing to do with the mechanism of action of the drug.
Absolutely, I totally agree. And in fact, that's why integration of this pharmacokinetic information into ReFRAME is important for us. I mean, in the world of where we're using acoustic dispense technology, there's nothing that should stop us from providing the primary screen to be done at a physiologically relevant concentration to begin with. It's just
the second thing is that the 1,000 reporters that they have, they can predict the other genes at equilibrium when you achieve steady state, but not after a perturbation.
Yeah.
And so the advantage, so if you want, the only reason I'm mentioning this is because we would be happy to provide the data. We would have to see which compound overlap between what you've screened and what we have, but we've basically screened every FDA approved drug in oncology and everything in phase 2 and phase 3.
Yeah, it would be great to do that. And I think, again, part of this exercise, both for the gene signature work, as well as other kind of what I call all this kind of information is kind of be foundational information on the compounds and relate that to the drug screening information. I think is critical. Where we're landing on this, what success looks like for us with this collection, with the work funded by the Gates folks, is to provide this to as many investigators as possible, in reasonable quantities, obviously.
So maybe we touch base offline and to see how we can share this data with you. I saw Barry Honig raising his hand, but now is no longer. So Barry, do you have a question?
Yeah. This is really a trivial question. I apologize in advance, but for those of us that do in silico work, is it easy to link to other databases? Each compound, is it just listed by name? Are there smiles or InchiKey? How easy is it to use and to link to other research?
Right. So Barry, thanks. So what we've been able to do is, for all of the hits that are defined as actually confirmed hits, like I showed, for example, in that Vero cell screen that we ran, all of those hits, the smiles and the related drug information is downloadable. So that can be available for every assay. What we are unable to do at this time, and again, it's a limitation of how the drug information is put together by the drug vendors, is that being able to get all 12,000 smiles or InchiKeys off of the ReFRAME portal is something we can't do right now, because the drug data vendor information that we have on the portal is in fact their data, right? So it's the source by which we generated, acquiring the compounds, et cetera. We have come to an intermediate agreement with those groups that we wouldn't be able to make all of the drug information, including the smile strings, available for any particular hit. So if you go through all of the screens that have been run on ReFRAME, all of the confirmed hits are all available for folks to download and be able to do follow-up in silico work.
And just quickly, I assume there's then no connection to the Zinc database. So,
not right now. So we've tried to incorporate some information from publicly available sources, including Zinc and PubChem, et cetera. We've put some of that into the portal, and especially some of the data, kind of the wiki data information is in there. We haven't done an at-large kind of linking event there. In fact, part of this linking event that we're going to do with this, agree on kind of the kind of links data set will be one of those connections we're going to make, but we're hoping to make some more between the compounds in this collection and the screening data here with what folks are can access in other places.
Thanks.